Hello and welcome students. Today I am going to deliver a lecture on genetic manipulation in animals and transgenesis in mice. Let's first try to have a look at the main objectives of today's deliberation. To give a brief idea about genetic manipulation of animals, explaining in detail the process of producing transgenic mice, to discuss the concept of pronuclear microinjection, to explain the process of transfection of embryonic stem cells and gene targeting in embryonic stem cells, to learn about designing targeting vectors and selection strategy. Finally, discussing about applications of genetically modified mice and gene targeting. Now we will start with the brief idea about genetic manipulation of animals. The genetic manipulation of animals has revolutionized our understanding of biology by making it possible to test gene expression and function at the whole animal level. Gene transfer techniques can be used to produce transgenic animals in which every cell carries new genetic information. Similarly, it is possible to introduce specific pre-selected mutations to the animal genome. The whole animal is the ultimate assay system in which to investigate gene function, particularly for complex biological processes such as development. Genetic manipulation in animals for higher productivity is called as genetic engineering. It refers to the alteration of the genes of an organism. It involves manually adding new DNA to an organism to add new traits. Examples of genetically engineered organisms include plants that are resistant to certain insects, plants that tolerate herbicides, and crops with altered oil content. Genetic engineering is the name of a group of techniques used to identify, replicate, modify, and transfer the genetic material of cells, tissues, or complete organisms. The objective of this technology is to increase disease resistance, productivity, and product quality in economically important animals by adding information of DNA markers to phenotypes and genealogy for selection decisions. Animals containing foreign gene are termed as transgenic animals. A genetically engineered or transgenic animal is an animal that carries a known sequence of recombinant DNA in its cells and which passes that DNA into its offsprings. Recombinant DNA refers to DNA fragments that have been joined together in a laboratory. Transgenics can be used for a variety of purposes covering both basic research and biotechnological applications. The study of embryological development has been extended by the ability to introduce genes into eggs or early embryos, and there is scope for manipulation of farm animals by the incorporation of desirable traits via transgenesis. The use of whole animals for the production of recombinant protein is a further possibility, and this has already been achieved in some species. The term farm animal or farming from pharmaceutical is sometimes used when talking about the production of high-value therapeutic proteins using transgenic animal technology. When considered on a global scale, the potential for exploitation of transgenic animals would appear to be almost unlimited. Achieving that potential is likely to be a long and difficult process in many cases, but the rewards are such that a considerable amount of money and effort has already been invested in this area. There are several possible routes for the introduction of genes into embryos, each with its own advantages and disadvantages. Some of these methods are: number 1, direct transfection or retroviral infection of embryonic stem cells, followed by introduction of these cells into an embryo at the blastocyst stage of development. Number 2, retroviral infection of early embryos. Number 3, direct microinjection of DNA into oocytes, zygotes, or early embryo cells. Number 4, sperm-mediated transfer. Number 5, transfer into unfertilized ova, and number 6, physical techniques such as biolistics or electrofusion. Now let's discuss another part of our today's lecture, that is generation of transgenic mice. In 1981, Jordan and Ruddle introduced the term transgenic in relation to mice genetically modified by transmission of foreign DNA into early embryos. Today, it is applied to the characterization of an animal whose genome has been altered by the stable integration of an in vitro recombinant genetic sequence. Three different methods are routinely used to generate transgenic mice. These are: number 1, transfection of early embryos with recombinant retroviruses carrying the gene of interest. Number 2, pronuclear microinjection of DNA into a zygote. And number 3, gene transfer into pluripotent embryonic stem cell originally isolated from inner cell mass of a blastocyst. In addition to these methods, the technique of nuclear transfer is sometimes associated with the transgenesis protocol. Now let us try to discuss some of these methods in detail. First in line is the pronuclear microinjection. The most popular approach for generating transgenic mice is the direct injection of transgenes into one pronucleus of the fertilized oocyte. This method can be carried out without the use of any specialized vector. The procedure involves the direct injection of transgenic DNA into the nucleus of the host cell with the help of glass microinjection tube or syringe. The instrumentation for microinjection consists of a microscope of high magnification attached with fine needles, syringes, and other facilities to carry out the delivery of DNA into the selected cells automatically. The injected embryos are subsequently transferred into the oviducts of pseudo-pregnant surrogate mothers. The pronuclear microinjection method of producing a transgenic animal is based on the introduction of linear DNA sequences into the chromosomes of fertilized eggs. The foreign DNA must be integrated into the genome prior to doubling of genetic material that precedes the first cleavage in order for the animal to be born with a copy of this new information in every cell. For several hours following the entry of sperm into the oocyte, the male and the female pronuclei can still be seen individually under the light microscope, and they have not fused yet into a so-called zygote. The foreign DNA may be injected into either pronucleus with no difference in results. However, the DNA is typically injected into the male pronucleus because it is slightly larger and closer to the oocyte surface. These oocytes are subsequently transferred into the uterus of pseudo-pregnant recipient animals. The offspring is screened to confirm a successful integration of the gene of interest for use in further studies. Next is the transfection of embryonic stem cells. Embryonic stem cells are derived from the inner cell mass of blastocysts. These cells are pluripotent, which means that they can develop into almost any type of tissue. Embryonic stem cells are used for more precise modifications of the mouse genome. This technique makes it possible to insert as well as remove or modify DNA sequences. Knockout, knock-in, and conditional mutant mice can be produced with this method. The first step is the removal of embryonic stem cells from the blastocyst. After transfection of the embryonic stem cells, selection, cloning, and screening methods make it possible to detect embryonic cell clones that demonstrate the desired site-specific recombination. After microinjection of the genetically modified embryonic stem cells into blastocyst stage embryos, the embryonic stem cells divide and become part of the embryo. The following chimeric animals will subsequently transmit the recombinant genotype to their offspring if the embryonic stem cells have contributed to their germ cells. Now let us discuss about gene targeting in embryonic stem cells. Gene targeting is a technique utilizing homologous recombination between an engineered exogenous DNA fragment and the genome of the mouse embryonic stem cells. Recombination between identical regions contained within the introduced DNA fragment and the native chromosome will lead to the replacement of the portion of the chromosome with the engineered DNA. These modified embryonic stem cells can then be injected into mouse blastocysts, where they can incorporate and contribute to the fetal development along with the blastomeres from the inner cell mass. These techniques can be used to ablate or knock out gene function throughout the mouse in selected tissues or at specific time point of mouse development. They can also be used to introduce mutations into the genome at a desired location. Essentially, all gene targeting experiments have the following steps. Number 1. Construction of targeting vector containing regions of identity with the mouse chromosome, a selectable marker, and planned modifications that ablate or alter the expression of the targeted gene or region of chromosome. Number 2. Introduction of linearized targeting vector into mouse embryonic stem cells and selection and screening for the targeted embryonic stem cell clones that have integrated the planned modifications by homologous recombination. Number 3. Micro-injection of targeted embryonic stem cells into blastocysts to generate mice chimeric for the targeted embryonic stem cells and host blastocyst cells. Now let's discuss another part of our today's lecture, that is designing targeting vectors. The generation of mutant mice by gene targeting takes the advantage of remarkable ability of embryonic stem cell lines to participate in the formation of germ cells of mice when the cells are put back into early embryo. Cell lines which have undergone gene targeting are enriched by the incorporation of selectable markers into the targeting vector. Out of this enriched set of cell lines, the desired homologous recombination event is identified by molecular analysis of genomic DNA. Targeted embryonic stem cell lines which have a normal number of chromosomes are identified and selected to make chimeras. Targeting can be used to generate knockouts, knock-ins or conditional alleles. Knockouts are used to define the overall requirement for a gene and to model loss of function mutations. Knock-ins introduce heterologous coding sequences such as reporters or DNA recombinases, or to incorporate changes in DNA sequence to humanize a mouse genome or to generate point mutations. Conditional mutations are used to define the organ, tissue or cellular autonomy of mutant effectors to circumvent embryonic lethality or to model somatic mutations. The two major hurdles for the generation of gene-targeted mice are obtaining homologous recombination and germline transmission. Although it has been almost 20 years since the first gene-targeted mice were constructed, not all parameters which affect the frequency of homologous recombination and germ cell transmission are known. Now let's try to learn about selection strategy. Gene targeting and transgenic technologies have different strengths and weaknesses with respect to fidelity to the disease, minimized variation and avoidance of unintended consequences. The generation of genetically manipulated mice for any purpose involves significant time and expense. This investment is amplified in the case of models of dementia because of the aging component in many dementia models. Investigator has to make wise choices in selection of technologies such that unwanted variation is minimized and fidelity to disease can be maximized. This is not a cookbook of how to perform gene targeting or transgenics aimed at transgenic or targeting cores, but is intended to aid the investigator in making decisions about which approaches are best suited for achieving their experimental goals, while highlighting some of the pitfalls associated with different approaches. Consideration must be given to the genetic mechanism of the disease to be modeled. Typical genetic mechanisms of disease include mutations which eliminate gene function, mutations which alter gene activity, and mutations which increase gene copy number. If a disease results from complete loss of function, that is a null mutation, then the gene can be inactivated by gene targeting. Both homologous recessive traits, that is both gene copies inactivated by mutation, and haploinsufficient traits, that is one gene copy inactivated, can be modeled by gene knockout. If a disease results from increased or novel activity of the gene product, then either transgenes which express mutant product or targeted sequence changes to the endogenous gene can be used. If the mutations lead to partial loss of function, then gene targeting can be used to introduce a specific compromising mutation into endogenous gene. Also, consider that while many disease-causing mutations are not complete loss of function mutations, that is null mutations are the most informative mutation for the normal function of the gene and can be used in conjunction with the disease-causing mutations to better understand molecular mechanisms. Now let's discuss another part of our today's lecture, that's applications of genetically modified mice. Transgenic mice, that is mice containing additional transgenes as opposed to those with targeted mutations, have been used to address many aspects of gene function and regulation. A vast literature has accumulated on this subject and genes concerning every conceivable biological process have been investigated. Besides their use of basic scientific investigation, transgenic mice can be used for more applied purposes such as models for human disease and the production of valuable pharmaceuticals. Many mouse models for human disease have been generated by gene knockout. For example, much information concerning the pathology of prion disease has arisen from the study of transgenic mice expressing mutant prion transgenes. Transgenic mice expressing oncogenes have been extensively used to study cancer. Genetically modified mouse models in which a specific gene is removed or replaced have proven to be powerful tools for identification of target gene and scientific understanding of molecular mechanisms underlying drug-induced toxicity through mechanistic studies. In spite of the advantage, there are significant limitations of genetically modified mouse models. Modification of a given gene does not always result in the anticipated phenotype. In some instances, phenotypes of targeted mouse mutants were not those predicted from the presumed function of the given genes, while other null mutants revealed no apparent defects. Furthermore, the phenotype outcome can be influenced by many environmental and genetic factors. Therefore, interpretation of the significance of the findings from the studies using genetically modified mouse models is not always as straightforward as one would expect, especially when desire is to extrapolate the findings to humans. Interestingly, many humanized mouse models have been generated for evaluating the function and regulation of cytochrome P450 enzymes. Our fascination with humanized animals dates back to ancients. For example, the Great Sphinx of Giza, a large half-human and half-lion statue, is believed to have been built by Egyptians about 4,500 years ago. Although the creation of humanized animals that carry a particular human cytochrome P gene provides useful tools for scientific understanding of the function and regulation of the cytochrome P enzyme, these humanized mouse models are not so useful in prediction of human pharmacokinetics in quantitative sense. Accordingly, it is important to keep in mind that an animal engineered to express a human disease and its protein is still an animal. Now at last, let's learn about application of gene targeting. Gene targeting has been widely used to study human genetic disease by removing, that is knocking out, or adding that's knocking in specific mutations of interest to a variety of models. Previously used to engineer rat cell models, advances in gene targeting technology are enabling the creation of new wave of isogenic human disease models. These models are the most accurate in vitro models available to researchers to date and facilitating the development of new personalized drugs and diagnostics, particularly in the field of cancer. Gene targeting is a powerful tool to dissect genes' functions. Recent developments of recombinating and site-specific recombination have dramatically increased the efficacy of gene targeting. In last decade, there has been a huge influx of data from gene targeting approaches to airway diseases. The approaches greatly help us to understand the mechanisms from signal transductions to immune response in airway diseases. Recent human association studies revealed many novel genes underlying complicated airway diseases. The next challenge will be the systematic analysis of the precise functions of these genes in the pathogenesis of the diseases. That's all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.